Okay, welcome to the main session of the afternoon. This session is entitled Harnessing Big Data to Understand Women's Mental Health. I'm Aristotle Vaneskis. I met you all yesterday morning, and I'm personally very excited about this panel because I love big data. I will make one comment. I hope it's okay, Lisa, which is that I think in order to do great sex and gender specific analyses, you need good sample sizes. And so in order to do that, you need a good overall sample size. And so I hope that we're going to see some great sample sizes here and some adequately powered studies to answer the critical questions of the field. Okay. I'm fierce about speaking time, so speakers watch out. I'm kidding, slightly. I'll stand up, you'll notice. And we have four fantastic speakers who come to us from all over. Just going to quickly list their names, then I'll introduce them before each. So we have Dr. Claudia Barth from, now I've forgotten what she told me, but Diakonhjemmet Hospital, I think that's okay, in Oslo, Norway. Dr. Elvisha Dhamala from the Feinstein Institute for Medical Research in New York, Dr. Emily Jacobs from UC Santa Barbara, and Dr. Marianne Seney from the University of Pittsburgh. So super excited to have Dr. Barth do the first presentation, and she's going to get us going. Right.
Hi, everyone. Thank you, Lisa, for inviting me. My original title was The Aging Female Brain, the Role of Immunoendocrine Factors During Pregnancy and Menopause. But after I saw that I was placed in the Harnessing Big Data panel, I really thought, let's switch it up and change the focus. I will still talk about immunoendocrine factors, at least a bit. As you all know, I think it has been presented very eloquently during the last two days. There are prominent sex differences in the risk of depression and Alzheimer's disease, as depicted here, with higher rates in females than males. And these disease risks fluctuate across the lifespan, paralleling hormonal changes. Here, for instance, in red is estradiol, the most potent and prominent estrogen in the female body. And estradiol fluctuates over the course of each menstrual cycle. And if pregnancies occur, it increases during pregnancy and declines postpartum. And it further declines during the transition to menopause. These major shifts in estradiol can predispose females to mood disturbances, as has been nicely discussed before, with a heightened risk for developing clinical depression, such as premenstrual dysphoric disorder, peripartum depression, and perimenopausal depression. While estradiol seems to protect females from cognitive decline during reproductive years, low levels of estradiol after menopause have been associated with females' increased risk for Alzheimer's disease later in life or in older age. Precisely how estradiol, and for that matter, other prevalent sex hormones affect females' vulnerability for affective and neurodegenerative diseases is still very much a matter of debate, because as been highlighted throughout, it's not that easy to study. And I want to make the case that actually, to be able to study that, we need three main things. We need granularity, scale, and methods. And I think there have been very nice examples of granularity during today and yesterday. And for my talk, I actually really want to focus on scale, by how can we harness big data, and by methods. How can we use machine learning and causal frameworks like Mendelian randomization to approach this topic? And when it comes to scale, as you might know, the UK Biobank is quite exceptional. If you do not know, the UK Biobank is a prospective population-based cohort from the United Kingdom, which covers 500,000 participants. And it has a wealth of data with relevance to health outcomes. It covers physical measures, lifestyle measures, cognition and hearing, biomarkers, imaging, online surveys, physical activity, genetics. Like if you go into their UK Biobank browser, it's like humongous what they have. And they basically have all the omics you can think of, which is amazing. But what is also very neat about this population-based cohort, that the age at baseline is 40 to 70 years of age. Normally, aging cohorts and prospective aging cohorts kind of tend to be around 65 years old at baseline, rooted in retirement age. So having actually this age range allows us to study reproductive aging in females without actually missing the critical midlife window. For today's talk, I will mainly talk about how we harness their imaging and genetic data and their female-specific factor. But what is also important to highlight, the UK Biobank, as amazing as it is, it was not designed with female health in mind, and I will get to that later as well. In my research and the research my group does, we mainly focus on using this data set to figure out how female reproductive factors impact the aging female brain. And we particularly focus on these factors listed here. And to actually look at brain aging, there could be tons of ways. We kind of specialized in using machine learning for brain age prediction as a proxy of brain aging. And herefore, we use the so-called brain age gap, which is the estimated brain age based on a normative population derived from neuroimaging data. It can be T1-weighted imaging data, diffusion-weighted imaging data, kind of is a mix of flavors based on what you want to look at. And so it's the brain age gap is this kind of the brain estimated values minus chronological age. So for instance, if the chronological age of a participant is 50 and the predicted brain age is 45, we have a brain age gap of minus 5 years. So we have a so-called younger looking brain or younger brain age. Vice versa, if we have a chronological age of 50 and a predicted brain age of 55, we have a brain age gap of plus 5 years, so-called older looking brains, just to get you up to speed what I mean with this when I move forward. Utilizing this approach, really spearheaded and led by Anne-Marie de Lange, we found that pregnancy and the number of childbirth actually leads to lasting brain changes, which can still be traceable decades after females have given birth. And we found that a history of childbirth was both associated with younger brain age using T1-weighted data, like looking at gray matter, but we also found that there was like younger brain age with parity when it comes to white matter. Interestingly, it's not a very robust effect across modalities, but we also found that there seems to be a slight nonlinear trend. So there's younger brain age with 2 to 3 children, but it kind of seems to taper off with grand multiparity, where you have, again, this kind of slight shift to older brains with grand multiparity. But here it's also grand multiparity is a fairly small sample within the whole UK Biobank, but we do have some outliers, which I like to say as like we have some individuals who have given birth to 22 children. So that is pure multi-grand parity. But we, interestingly, the effects were most prominent in the basal ganglia and the limbic structures. And we just published a paper this year where we extended this framework to fathers, and we could also find younger brain age in the fathers, but the effects were less prominent, and the effect we found in the basal ganglia and limbic structure was only limited to the females and were not detectable in the males. Besides the effects of previous childbirths on the brain, we also examined the other factors I've previously shown. Here, for instance, we looked at an index of cumulative estrogen exposure. We looked at HIT user status and oral contraceptive user status. And we found that oral contraceptive user status was not associated with brain aging in this cohort, but we found older brain age with HIT use, and we found also older brain age with a higher index of cumulative estrogen exposure. And in a later, like in the newest study from a PhD candidate in my group, Ariel Crystal, we could also find that age at menopause seems to be linked to brain aging by particularly like older age at natural menopause seems to be linked to younger brains, but that's not the case if the menopause was induced surgically. So extending, like besides assessing the effects of female-specific factors on the brain, we also looked at these factors on cognition. And here we found, which kind of hardly mirrors the effects we've seen with brain aging, but also not really, which is kind of confusing, but interesting. Here we were able to utilize basically the whole power of the sample. So the sample size is 221,000 individuals. And here we found that the longer reproductive period, older age at menopause, and oral contraceptive use were positively associated with cognitive performance in late life. And number of live births and HIT use actually showed mixed effects, with task-specific positive and negative. Or like negative is also a negative association. For instance, for childbirth, we found better performance scores at simple processing speed and visual memory, but we found slightly kind of lower scores when it comes to complex processing speed and executive functioning. Similar effects were seen for HIT use. What I want to kind of highlight that the effect sizes in all of our studies are fairly small. It's really important to highlight that, and I think it has been highlighted in previous talk, that we do not overinterpret what we find. But what I did find interesting in this particular study was that the effect of oral contraceptive use on cognition was fairly large even. And I find it particularly interesting because, again, it's a prospective aging cohort. They were 40 to 70 at inclusion. So this effect might not replicate because these women were taking very different oral contraceptives than are on the market today when it comes to dosage and formulation. But again, the HIT effect kind of has been sticking out a bit. And as some of you might know, when it comes to HIT use and the brain, that's a very debated field of research, especially after the Women's Health Initiative from 2003, finding negative effects on the brain. Sorry, I'm recovering from a cold, and when I speak long, I start coughing. But what I want to kind of get at is that we did kind of a follow-up study where we really wanted to do a deep dive into the HIT effect, where we kind of wanted to use UK Biobank, but also pull the prescription registry data from the UK to see actually what kind of HIT were they using. Here we used the biggest sample so far of nearly 20,000 women with imaging data. So the first study we did on that from 2020 had 16,000. So we could utilize a new release of data, and we stratified the users by never users, current users, and past users. And interestingly, the previous effect we saw was specific to the current users and was absent in the past users. So in the current users, we saw older gray matter brain age, older white matter brain age, and smaller hippocampal volumes. We did not see an effect on white matter hyperintensities, which is a marker of vascular health. We also saw that older age at last HRT use and longer duration of use, particularly post-menopause were associated with older brain age gap and lower hippocampal or smaller hippocampal volume. Interestingly, so these findings here, we excluded everyone with a hysterectomy and ovarectomy. So we really looked at like healthy users. But when we kind of compared MHT users, so HIT users, MHT, you also see in our papers, we went from HIT to HT to MHT to be more precise as we learned the language which is most appropriate. But what I want to highlight here that we actually found younger brain age and higher hippocampal volumes in the MHT users with surgery relative to the NHT users without surgery, which is very interesting. As we saw yesterday, there are many menopauses and normally surgery is associated with worse outcomes. But here we see actually relative to users without surgeries, it might actually be better. One reason for that could be that MHT use is much more tightly linked to surgery than if surgery does not happen. Normally, MHT is given if menopausal symptoms occur. I'm so sorry. And when menopausal symptoms occur. But that can also, there can be quite a delay until between like when symptoms start and when women actually receive treatment. If a surgery happens, this is much more time locked. And that might actually be beneficial for brain health. Coming to the association between different MHT regimes and brain measures. So we did a deep dive in the prescription registry data. Unfortunately, relative to the Scandinavian prescription registry data, the UK is quite scattered and it's not as beautiful. So it has its difficulties. But for roughly 538 individuals who had the imaging data and oh, I'm very late with my coughing. So to speed everything up, is like we found actually no associations between different MHT regimes and brain measures. If it's estrogen and progesterone, or if it's estrogen only route of administration, active ingredients, we did not find any association between these different regimes and brain measures. Before correction for multiple comparison, we did find indication for a negative effect of oral use and actually of CCA and like kind of combined use preparations, which is in line with previous findings, but again, this did not survive correction. So as an intermediate summary, we find indication that older age and menopause and longer reproductive span is can be beneficial for cognition and potentially brain aging. Hysterectomy and bilateral ovarectomy, when it comes to MHT use, seems to have a different effect than MHT use for other implications or indications. Number of childbirths has been associated with younger brain age, and all contraceptive use showed like high kind of very strong impact on cognition, but we didn't find any impact on brain aging. And when it comes to HIT use, we do find like older brain age in the current users, but the story is fairly complicated. So I really want to kind of bring home that we do not find like proof for neurotrophic effects, but we also do not find any proof, very strong proof for any adverse effect. It is complicated. What we also were wondering, like, these are observational cross-sectional approaches. So what can we say about causality? We can't really say anything about causality. So we really wanted to kind of move from an observational to a causal framework. And for that, we applied Mendelian randomization, which I now just appropriate as MR, to test for causal effects of estradiol and female reproductive factors on brain and mental health. MR is a statistical approach that uses genetic variants to probe the association between a genetically predicted outcome, for instance, estradiol levels, and a genetically predicted exposure, like estradiol levels, and genetically predicted outcome, like brain aging, or in this case, also risk for Alzheimer's disease and depression. As the combination of genetic variants a person receives at conception is presumably random, that's what we assume, MR can be interpreted as a natural version of an RCT, a randomized control trial, and is therefore much less prone than observational studies, which I presented before, to kind of confounding and inverse causality. Because I'm very slow with all this coughing. I try to speed that up because the findings are kind of slightly underwhelming. This work was done by Hannah, my PhD student. So she did amazing work. Oh, and the slides are a bit off, but she did amazing work looking at genetically predicted estradiol levels alone and accounting for BMI, and looked at how, if there's a causal link to brain age gap, depression, recurrent depression, and Alzheimer's disease. And she found, no, there is no causal link. She also did not find any causal link when she accounts for BMI, and BMI was also not causally linked to any of these outcomes. Then she looked at so-called proxies of estradiol exposure. We like to call them like that, like the other female reproductive factors we've been talking about. Age at menarche, age at menopause, length of reproductive years, number of childbirth, HRT use, or contraceptive use. And again, nothing. There was no causal link with any of these outcomes. The only thing she found was actually that age at menopause seems to be causally linked to depression. So a younger age at menarche seems to be causally linked to a higher risk of depression. But this finding is not robust across all available MR methods. So we're a bit cautious about this effect. So in summary, 0 minutes left. Observational studies suggest that reproductive history and hormone use might influence brain aging cognition later in life. But causal frameworks do not suggest a causal link between estradiol levels and female reproductive factors and brain and mental health. So I'm not going into that. What could be a factor? And I want to give a shout out to this perspective piece we wrote, trying to really tie in the immune system. And when it comes to scale, big data is great, but currently it's not designed for women's health in mind. I think Emily Jacobs will talk about that, and I really just want to kind of shout out our newly launched Enigma Neuroendocrinology working group, where we really try to combine global already available data on these topics to really boost power, reproducibility, and generalizability by pooling data sets across the globe. If you want to learn more about it, we just got our nice commentary published, and we have a website, so you can reach out. And we also have a call for papers for, if you have any papers on sex and gender and neurodegenerative diseases, reach out. You're very welcome to submit. And thank you. Birgit highlighted the Lawrence workshop. That's the only picture I have of me and my PhD students, which Lisa took. I feel that's very telling. So I'm very sorry for coughing through it. I felt good until I had to talk. So yeah, please ask questions, reach out. And if you want to contribute to our efforts, please reach out. Everyone is welcome, particularly the ones with data.
Claudia is ready to cede her time to the next speaker, but we have time for one question. Okay, whoa. Okay, I think that was the first hand I saw, very back. If it's very quick, then we can maybe squeeze a second one in.
Hi, fantastic talk. I was particularly interested about the randomization, the Mendelian randomization at the end and how it seemed to, estrogen directly doesn't seem to have an effect. Do you think it could be maybe related to another factor linked to ovarian function, such as like ovarian reserve or like your anti-Mullerian hormone levels? Because I've seen some associations with that with cardiovascular health. So I thought maybe it could be an indirect, maybe there's something there.
I think like the estradiol finding I felt was not too surprising because we had heard amazing talks about like that it's mainly the brain's, most likely brain sensitivity to these fluctuations, not the absolute levels, which seem to be like causal or have an effect. What else it could be is interesting because I was very surprised that we didn't find any causal links between the female reproductive factors besides H-Menarche, and the H-Menarche association was quite weak. So one step we definitely want to look into inflammatory systems and immune markers and use genetically predicted measures. But we also kind of really want to combine that with the granularity approach I presented at the beginning, having like really deep phenotyped samples where we have all these measures and try to actually see longitudinally what like happens to disease risk and the brain, for instance, across perimenopause. So cross your fingers, I get the money, then we can do that. But I think what I want you to take home from that, it's like, it is complicated. There's no simple causal link between these things. So there might be very different things under the hood happening, and we really want to figure out what it is, because we have all these like association studies, which are very great. We contributed a lot of them too, but we really want to also kind of, in the long term, try to help people and have actually kind of a predictive framework. So we really need to kind of better understand and also get better samples to be able to understand these things. And yeah, so I really want to say like granularity, scale, and methods moving forward will be really important to kind of move this field forward.
Thank you very much. So Dr. Elvisha Dhamala next from the Feinstein Institute for Medical Research.
Like, I hear you.
Hi everyone, I'm Elvisha, and today I'll be talking about how we can leverage brain-based predictive models to better understand women's health. So I want to start off by first thanking Lisa for putting all of this together, everyone who contributed to this, and everyone who's been presenting, listening. It's been so amazing just learning about all the really cool work that's going on in this space. And I hope that I can present something that's slightly meaningful. So the good thing about being right now is that I don't have to do any of my intro slides. So I'm going to skip through this, but basically when I talk about sex, I'm going to be referring to sex assigned at birth. And when I talk about gender, it's going to be referring to attitudes, feelings, and behaviors that are associated with a particular sex. Goes without saying, but sex is not the same thing as gender, but there has often been a conflation for many different reasons, including the one that was mentioned yesterday. In some languages that the distinction between sex and gender just doesn't exist, but in other cases, it's also just been conflated with one another. But it is important to recognize that both sex and gender are multidimensional constructs that are influenced by different aspects of the brain, biology, environment, sociocultural factors, and so on. And sex differences in the brain, as we typically understand them, and in behavior, may be driven by a number of sex and gender-related factors. So these may include things like hormones, chromosomes, the HPA axis, societal norms, gender identity, stereotypes, as well as individual experiences. And it's important to recognize that just because there are these differences does not mean that one sex is inferior to the other. All it means is that it's different. Different does not mean better. It does not mean worse. It just means different. Just want to make that very clear because I'm going to be talking about a lot of these differences. So when we start to talk about these differences, There are a lot of sex differences in the prevalence of neurodevelopmental disorders. So we've heard a lot about different ones that in the last couple of days. So this includes particular differences in things like autism spectrum, ADHD, and conduct disorders, which tend to be more prevalent in males than in females. If we look at neuropsychiatric disorders, there are again sex differences. Here, typically depressive, anxiety, and eating disorders tend to be more common in females, whereas schizophrenia and substance use disorders tends to be more common in males. And finally, there's also sex differences in the prevalence of neurodegenerative disorders, where they're a lot more prevalent in females, particularly Alzheimer's. And when we start to think about these brain disorders, so whether it's during development, psychiatric illnesses, or degeneration, we have to think about how they might be related to the brain and in which ways the brain might actually be underlying or in some part contributing to these disorders. So to do that, we look at often different aspects of brain structure and function. And in the brain, there are white matter connections that directly and indirectly connect different brain regions that facilitate the flow of neural activation signals. Together, these structural and functional properties of the brain underlie our unique and individual behaviors. If we start to look at the brain, there are critical sex differences in brain structure. So throughout the lifespan, the male brain is about 10% larger than females, and these differences in structure exist in gray matter volume, white matter volume, subcortical gray matter volume, as well as ventricular volume. There's also notable differences in brain function. So specifically, there are differences in the types of within and between module connections that exist in males and females. And it's not just that, there's also differences in how the brain is functionally organized. So if you look at the functional brain networks that exist, they can actually be used to distinguish between males and females. And the networks that are particularly important are ones that we call heteromodal association networks. So these include things like the ventral attention, default mode, frontoparietal, and dorsal attention networks. So all that to say, sex has a very strong influence on the brain, and there are these differences that we have to acknowledge. But not all sex differences are the same. A fundamental assumption of my research and a lot of brain research in general is that there is something in the brain that is somehow in some way associated with some aspect of behavior. And a lot of times we're looking at these differences thinking these differences are all going to emerge or exist in the same way. But sex and gender-related factors may influence aspects of the brain, the behavior, or the association between the two. And they're very different in what they mean and what they represent. So in my research, I use these brain-based predictive modeling techniques to look at these differences in brain behavior and brain behavior relationships. So typically, when we've studied differences across the sexes or across genders, we've looked at them at a group level to compare between males and females, boys and girls, men and women, and so on. But everything has been done at a group level without taking into account individual variability that may be driving these differences. Now, with increased access to big data and computational resources, we can actually leverage what we call brain-based predictive modeling approaches to understand how neurobiology underlies behavior and take into consider the insights from the group level analyses and then use an individual's own neurobiology to make predictions about their own behavior. And with these techniques, we can perform what we call descriptive, predictive, and prescriptive analyses. So descriptive analyses rely on existing observations to identify underlying patterns to describe and summarize the data in an easily interpretable manner. In other words, you can use this information and these modeling approaches to generate, for example, data-driven biological subtypes of disorders. We can also do predictive analyses where we establish associations between independent and dependent variables in existing data and use those associations to predict trends and properties in unobserved samples. So in other words, using these, we can learn brain behavior relationships and then use an individual's own brain to predict how they might have specific cognitive abilities, personality traits, specific mental health diagnoses, or dimensional symptom profiles. Finally, we also have prescriptive analyses, which enable us to recognize risks and offer potential treatment solutions. So in these types of analyses, we can actually see, based on a person's brain and history of other data that we have, is a person likely to respond to a specific therapeutic intervention, or is there something else that might be better for them? And collectively, we can use these descriptive, predictive, and prescriptive analyses to yield clinically meaningful insights to facilitate the development of personalized diagnostic tools as well as therapeutic interventions. But when performing these analyses, there's a critical factor that we have to consider. And it's not just one, but it's many. So specifically, we have to consider how different methodological choices can actually affect these models' performances and their interpretabilities. So different choices that play a role are things like the neuroimaging modality and the state that we choose to base our predictions on, or these models on, the parcellations that we use to segment the brain, how we transform our data before it gets put into the model, the sample size that we have, whether we're using population-specific or general models, the algorithms that we're using, so machine learning versus deep learning, and so on, and also the specific phenotypes that we're trying to predict. And today, I'm going to specifically be talking about how we can leverage population-specific models to better understand male and female-specific brain behavior relationships to advance our understanding of women's health. So it's a very straightforward concept. Take all your data and separate it. I'm not doing anything that's too crazy, but what we can do is using this population-specific modeling approach. A lot of times what I've done is I've simply, like I said, separated the data by sex, but you can also separate them by other groupings. And using these groupings, we can then develop models that are specific to each group and then see if those brain behavior relationships that we capture are the same across these different groups, or if there are any differences. We can also see if models trained in one population generalize to the other, and so on. So with that, I'm going to talk about a few different studies. And the first one that I'm going to talk about is actually one where we use functional MRI data and the functional connectivity that was extracted from that to see how sex and gender influence the brain. So I've shown you before, there's a lot of work that's shown that there are sex differences in the brain. We wanted to see if we can take it a step further and also see if gender, independently from sex, influences the brain. So I should mention that all of the data that I'm going to present, all these analyses were done in the ABCD dataset, which is the Adolescent Brain Cognitive Development. The study had a sample size of about 4,600 children, all between the ages of 9 and 10. So just looking at sex differences in the gender identities, what we see is that males report stronger sex-congruent gender identities than females. So in other words, people assigned male sex at birth are more likely to align with that gender than people assigned female sex at birth. And then using the functional connectivity data, we wanted to see if we could predict sex as well as the gender data. And we had both self-report and parent report data. And there's a little asterisk at the top because this is slightly misleading. So here we trained the model on everyone, and we were able to explain about 60% of the variance relating to sex, as well as about 58% relating to gender. But here, because this model includes everyone, obviously the gender predictions are going to be influenced by the sex itself. So we use population-specific models or sex-specific models to then see if we could predict gender in these individuals. And we can predict gender identity derived from these parent report scores better than chance, but we can't do it with the self-report scores. What might be happening is the self-report scores have a much smaller scale. It's only 4 questions, and the distribution is very skewed. Whereas if you look at the parent report, it actually captures a much wider range of information relating to gender. So we think that might be explaining it. But it's really interesting to see that even once you remove that effect of sex entirely, gender itself has an independent influence on the brain. And this is emerging in kids as young as 9 years old. So you can only imagine that throughout the lifespan, as we're exposed to more and more gendered experiences, it's not just sex differences that we're capturing when we look at sex differences, it's also gender-related influences that need to be considered separately. So then we looked at what are the actual functional networks that are associated with sex and gender. I'm not going to go through this in too much detail, but basically the correlates of sex are different than the correlates of gender. So let's just talk about what those are. And if we look at the brain networks that were involved in the predictions of sex, what we see is that there are kind of like dispersed connections throughout the brain that are involved, including both unimodal and heteromodal association networks. And a lot of it is kind of centered around control, limbic, some of the attention, but also somatomotor and visual networks. Next, if we look at the correlates of gender, they're actually pretty different from those for sex. So this is for the parent report data, assigned females on the left, assigned males on the right. And what we see is that in females, it's more localized within the temporal parietal and sort of like the default mode. on the attention networks, whereas in males, it's a little bit more dispersed, suggesting that not only is gender exerting an independent influence on the brain than sex, but that independent influence might also be different across the sexes. So this now tells us that when we're thinking about how sex or gender might be influencing the brain, or it might not, it's very critical to consider them in these models, because if these factors independently are influencing the brain, to such a strong extent, then it makes sense to say that it's probably also going to influence other brain behavior relationships that we're interested in. So I have 7 minutes. I'm gonna go through this really quickly. So next, we wanted to look at how does sex actually influence relationships between the brain and the expression of internalizing and externalizing behaviors in males and females. Again, same data set, ages 9 to 10. I made this pretty picture for a figure, but the editor didn't like it, so you guys get to see it. So we had about 5,200 kids in this study, again, also from ABCD, and we were using the functional connectivity data to predict aspects of internalizing, externalizing, as well as thought, attention, and social problems. And we used both syndrome scales as well as DSM-oriented scales, but I'm mostly going to focus on the internalizing and externalizing domains from the syndrome scales here. Looking at the overall expression of behaviors, we basically see that both males and females are exhibiting these types of behaviors in childhood. So it's not an inherent sex difference in the fact that one sex is expressing these behaviors and the other isn't. So that is not something that would potentially drive these results. We also see that they are generally similarly associated with one another or similarly correlated with one another in terms of these behavioral expressions, but there is a lot of variability. So again, it's not just that all of these behavioral expressions are inherently correlated, which might explain why our predictions would or would not generalize across the sexes. So looking at our model performance itself, we see that basically our brain-based predictive models can accurately predict psychiatric illness-linked behaviors. And specifically in males, we're able to predict all aspects of externalizing behaviors. So this includes the summary scores, as well as the rule-breaking and aggressive scores, as well as attention, social, and total problems. In females, it's largely similar, but we are also actually able to predict an internalizing behavior, and specifically withdrawn depressed traits better than chance. So then we wanted to see, can you take the model that's trained in males and generalize it to females and vice versa? These are our results. There's a lot going on here, but I'll go through each quadrant at a time. But on the vertical axis, we have the models, the behaviors that the models were trained on. On the horizontal axis is the behaviors that the models were tested on. Models trained in males are at the top. Models trained in females are at the bottom. Models tested in males are on the left. Models tested in females are on the right. And everything that I'm showing here with as per that color bar, so basically any box that's filled in, survived significant corrections for about 1,100 something comparisons across all of this. So it's very strict. So if we start by looking at the models that are trained and tested in males, what we see is that models trained to predict different types of externalizing behaviors generalized to predict other similar behaviors in males, as well as things like attention, social, and total problems. But what you'll notice is that those kind of dotted line or little boxes are models that were trained to predict externalizing behaviors and then assessed to see whether they generalize to predict internalizing behaviors. In males, that doesn't work. But when you take these same models that were trained in males and assess it in females, it does work. So what this tells us is that the same aspects of the brain that are underlying externalizing behaviors in males might actually be underlying internalizing behaviors in females. You can do the same thing the other way around. And here we have the same sort of plot, similar sort of trend with the externalizing behaviors. And then now the dotted boxes, dash boxes, whatever, are turned the other way around. So here it's models that were trained to predict internalizing traits and then evaluated to see whether they generalize to predict externalizing traits. So within females, when they're trained and tested, it does not work. But when you test it in males, it works. So again, this further provides evidence that the same brain networks might actually be underlying different sets of behaviors in males and females. I'm going to skip this. If we look at now the actual correlates of these behaviors, we assessed both positive associations, which are shown at the top, and negative associations at the bottom. And then the models for males are on the left, females on the right. And these were the correlates for rule-breaking behaviors. So overall, we see that across males and females, it's pretty similar. But if you now relate these to the correlates for an internalizing behavior, so with the withdrawn depressed traits, they're actually also the same thing. So again, it's the same set of brain networks, but how they are expressed might be in some way influenced by gendered, socially acceptable norms, and that might be kind of driving the types of behaviors that boys and girls, this is kids ages 9 and 10, are defaulting to. So this is telling us that gender, even at such a young age, is already having a very strong influence in how these illnesses might be presenting. So perhaps, I'm not a psychiatrist, but maybe we should rethink how they're diagnosed, how they're classified, and also how we try to separate these illnesses, because it might actually be the same illness that's just presenting as something different. So I only have 2 minutes, but I'm gonna go through this really quickly. We basically did the same set of analyses, but now we wanted to look at psychotic-like experiences. So these include a range of subclinical symptoms that resemble aspects of psychosis, but don't meet the threshold for diagnosis. So it includes things like perceptual distortions, false beliefs, and disorganized thinking or speech. And although these PLEs or psychotic-like experiences do not necessarily predict the onset of psychosis or schizophrenia later on, a lot of them actually, it has been shown that they are associated with the development of some mental illness later in life, suggesting that these might be a transdiagnostic early marker for risk of psychiatric illness. So in this sample, again, ABCD, 57% of males and 57% of females and 60% of males reported experiencing at least one or more PLEs, and males reported a greater total number, but there were no overall sex differences in experienced distress. If we now look at these brain-based predictive models in both males and females, we're able to predict both their total and their distress scores better than chance. So the true models are shown in green here and the null models in orange. And again, we wanted to see, is it the same or different brain networks that are underlying these predictions? There's some overlap, but there are also critical differences in these associations themselves. So if we look at the associations in females, which is shown in the left, and the associations in males, we see that they are actually quite different. So in females, there are dispersed functional connections throughout the brain that are associated with childhood PLEs, whereas in males, these experiences primarily map onto functional connections in limbic, somatomotor, and visual networks. So this now provides further evidence that these differences might explain why these markers might then indicate potential potential development of different mental illnesses later in life. They might also explain or serve as early markers that predict the type of symptoms or the type of psychosis that males and females are more likely to express later on in life. Skip this slide, but basically, why does all of this matter? If we consider sex and gender both independently and interdependently, it'll shed light onto how sex, gender, and related factors influence not just our behaviors, but also our brain development, brain organization, and the relationships between brain and behavior. And collectively, our findings suggest that the consideration of both sex and gender-specific biomarkers for different aspects of psychiatric illness risk is critical to better improve prediction, diagnosis, and treatment of these illnesses, which we all know exhibit critical sex and gender differences. So with that, just want to say thank you.
I think we have time for 2 questions. Okay, so one and then, well, that is a powerful hand back there. Okay, let's start here.
Thanks. So thank you very much. As I understand it, this was data from one wave of the ABCD?
This is only the baseline data.
Yeah.
Yep.
So my question, it was 9 to 10 years, or what about puberty? Did you incorporate that somehow? Maybe matter in the girls at least?
So here it's tricky because so something I should mention is everything here that was presented is baseline data except for the gender data, which was actually only collected at a 1-year follow-up. So then it became tricky to consider puberty, because then do you consider puberty at the 1-year time point, the baseline time point, and obviously in a year things will have changed. It's also tricky because already we were limited with sample size with our 5,000 participants, but within those participants, not all of them had puberty data. So if we were to try to maximize for participants that had everything, we were down to about 1,000 participants, which isn't necessarily going to generate reliable predictions here. So we have not but I do have interesting puberty results that I don't know how to make sense of. So if you do, I would love to talk.
Hi, first of all, thank you so much for your presentation. Very interesting area of research. So I'm very interested in the hierarchical taxonomy of psychopathology, like high top, and this really resonated with that for me. So I wanted to ask, for one of the initial models you've shown for sex, I think it was sex differences, not gender differences, and externalizing versus internalizing symptoms, may have been the very first figure. You mentioned that there wasn't a sex gender difference for these symptom behavioral clusters for internalizing versus externalizing. I'm just wondering if you included like substance use disorders and looked at an adult population instead, do you think this picture would be different and how would it change?
So are you referring to this? No, this one?
Yes, I believe so. Like yes. So you mentioned that there wasn't like a big sex difference, if I didn't understand incorrectly, for internalizing and externalizing symptoms. I'm just wondering if substance use disorders were included and if it was like an adult population, like would the clinical picture change? And because I feel like sometimes we can really see differences based on sex for these clusters, right?
Yeah, so these are kids 9 and 10. So they don't, as far as I know, have substance use disorders. But I think in an adult population, then definitely that's something that would be worth considering. And that's also somewhere where I would definitely expect to see differences in the brain correlates of those behaviors.
Thank you so much.
Very quickly, as Emily gets to the stage, question, we are way behind us, so I don't want to penalize the final two speakers.
I just had a quick question. Could you briefly expand on the gender scales? Because I'm just wondering what was captured. Like, it sounds like the child report one was gender identity, but it sounds like maybe the parent one was more about gender expression or like conformity to gender norms.
Yep. So the parent one was 12 questions. Everything's on a scale from 1 to 5. The child one was 4 questions. And like you said, everything was relating to felt gender. Whereas the parent one was related to gender expression. So a lot of sex type behavior, primarily during play, since they are 9 to 10 years old, or 10 to 11 years old at that point, as well as gender contentedness as reported by the parents. Yeah. So these data are going to be collected longitudinally, but the questions are going to change to account for the fact that the same questions at 10 years old will not be relevant at like 16, for example.
Right. And are you thinking of digging into those like smaller gender differences or just the scale as a whole?
Sorry, can you repeat that?
Kind of like specific items or subscales of the scale or just as a total score?
So here I was only doing the overall score for the self-report and the parent report.
Okay, we need to move on.
Thank
So Dr. Jacobs is next from UC Santa Barbara.
This feels like a family reunion. And like every good family reunion, there is the matriarch. So thank you for bringing us all together. There's like the kooky aunts and uncles, and then there's also like all of the new kids that you get to meet for the first time. And that's really what this feels like. It's been such a fun experience. So thank you. Okay, so before I dig into the science, I want us to take stock on the status of women's health. This is all preaching to the choir a bit, but it's, you know, the last session, so I figured we could have like a little movie to kick things off. Oh, do I have to unmute? Let's see. Okay, do I do this? No. You want to try? Oh no. Should I keep going or? Okay, okay. Yeah. I'll send it around, or Lisa can send it around later. But you know, I think the point of this video is to really put women's health in this moment that we're all in a historical context.
The male body has been treated as really the standard in science for centuries. This dates back to the time of antiquity, like when Hippocrates literally proposed that a woman's womb could wander around her body. And women were described as unknowable. And these same forces pervade our era, right? A time when biomedical research in animals is conducted primarily in males, and it took an act of Congress literally to require the enrollment of women into clinical trials. The British historian Eleanor Cleghorn says, "Our body is not elusive to us, but something about our body seems to thwart and frustrate medicine at every turn." That needs to change. And many of us in this room have come to the conclusion, right, that elevating women's health research is going to require a global shift in science culture. That shift starts now. It starts here. It starts with all of us using the tools and the talents and the resources that we have to push for change. This is all old news, but it bears repeating again and again and again, right? Women's health lacks funding. We are half of the population, but in the United States, only 10% of the NIH budget goes to conditions that primarily affect women. Members of this meeting, first and foremost, Lisa Galea, have done an enormous amount of research and advocacy to call attention to this disparity. And I am happy to report that with that advocacy, a shift has started. Just a few weeks ago, the president called for $12 billion in an executive order in new funding for women's health research. This couldn't come soon enough. As Lisa mentioned last night, the World Economic Forum in January put out a report saying that filling the gender data gap is going to unleash a $1 trillion return on investment. That's $1 trillion annually. So we literally cannot afford to wait on women's health. And because of peer pressure, I put pictures of the event in this slide. It was truly an honor and privilege of a lifetime to witness this watershed moment for women's health. So let's continue to put our foot on the gas. And we got a lot of work to do. And to put real numbers on the problem, I want to give one example from my own field of human brain mapping. So when MRI technology entered the mainstream in the 90s, it gave neuroscientists really kind of unprecedented access to the living human brain. Suddenly we have this tool for understanding how it works when it's healthy and what goes wrong when it's not. And since then, over 50,000 articles have been published on the human brain, represented by this blue wave. And then if you squint really hard, you'll be able to detect this thin black line at the bottom, as I call it. That's everything we know about how the brain responds to pregnancy or menopause, a menstrual cycle, hormonal contraceptive use, and so much more. So of those 50,000 articles, less than 0.5% focus on health factors that are specific or unique to women. So about 0.5% of the brain imaging literature. So there's a ton of work that we have to do to fill this gender data gap, which has been going on for 20 or 30 years. I hope it doesn't take that long for us to fill that gap, but we've got to put our foot on the pedal. So when we think about the scientific body of knowledge, let's ask, whose body does it serve? These videos or these pictures would make more sense if the video played. You know, I think to put it another way, neuroscience has overlooked aspects of the human condition relevant to half of the world's population. It's why we don't know more about what happens about birth control related depression or what happens to our brain during menopause, among other essential questions. And it's not that we can't find out, right? It's that historically so few have cared to, but now this room is going to be the answer to all of our woes, and we will begin to fill this data gap. So when we think about how the field is moving forward and what approaches I think we ought to take, I am starting to think about this from, again, this vantage point. I'm a human brain imager at heart. And Katerina Groton had a really nice article in Neuron a couple years ago where she's trying to take stock of some problems in our field. And part of that is limited sample sizes. And so she really made this pretty clear, cogent argument for two paths forward for human brain imaging. One that relied on consortium studies, so large N, the focus of this symposium, and another that was really focused studies that deeply characterize the brain to understand really kind of precision medicine. So how do brains over time change? That this is the kind of evidence we need to guide clinical care. Consortium studies can tell us about population tendencies, right? Really good at guiding social policies. So we need both of these roadmaps moving forward. And I'm going to tell you a little bit about how we're thinking about incorporating this roadmap into women's brain health. So I'm going to begin by just sharing a few examples of how dynamic changes in hormone secretion can influence the structural and functional architecture of the human brain. And so I'm going to follow this road to the right for a moment and focus on precision imaging studies from our lab that track the human brain with pretty high temporal precision, right, revealing the capacity for neuroplasticity. And it's not just my lab, Julia Sacker's group, Claudia Barth, like others have taken on this approach, and it's just revealing like super cool capacities for the human brain that I think have gone unnoticed, at least in the human literature for so long. In the animal world, this sort of thing has been done, but really not in humans. So I'll just share a few examples. Okay, so you all know this by now, but like a central feature of the mammalian endocrine system are these hormonal rhythms, right? They ebb and flow over time. These rhythmic changes in hormone production drive physiological processes throughout the body. But until recently, nobody really understood how they shaped the human brain, right? We knew they must be doing something because in rodents, they clearly are, but we didn't have the right tools or the right methodological approaches to really map how these endocrine fluctuations were influencing the human brain. Because cross-sectional studies that sample the brain like at a single time point just can't capture these dynamic transitions. But precision imaging can offer us a first glimpse, right? So this was one study that we launched during the pandemic because we were sitting around, we couldn't do our typical cohort study. So we said, well, what if we just scan ourselves? So Laura submitted herself to science. I call her the Marie Curie of neuroscience. She underwent brain imaging, venipuncture every single day, every 24 hours for 30 consecutive days, and then it did it all again a year later. She looked like a pincushion when it was over. It was all IRB approved, and she really did this herself. She had this perfect canonical menstrual cycle, right, with this changing in gonadotropins, change in estradiol and progesterone. So the question is, how do these endocrine rhythms influence the central nervous system? And we looked at this in a whole bunch of ways. You can see the publications at the bottom, some of them. The first finding was really just sort of correlational in nature, which is that as estradiol is peaking around the ovulatory period, you see this increase in functional connectivity. Maybe some, if we break it down into sort of the Elvisha style functional networks, we can see some networks are more sensitive than others. The morphological features of the brain are changing too. So if we use imaging of the hippocampus, we can see particular subfields that ebb and flow with progesterone. When we redid the experiment a year later, the participant was on a drug that selectively suppressed progesterone. So notice that this blue wave in the first panel is absent in the second panel. TLDR, that drug was the major form of oral contraception on the market today. So estradiol escaped suppression, but progesterone was suppressed, and these hormonal rhythms that we see are sensitive to progesterone were abolished on the pill. This is all open access data. I will say we also worked with OHSU. We have all of the levonorgestrel and ethanol estradiol data on this. So if people want to dig into the data, it's all there. There's been something like 20 publications now on 28andMe. So this is the power of open data. I do want to make one point, even if it's just a political one. These patterns are not exclusive to women, right? Diurnal changes in hormone production, that ebb and flow that occurs AM to PM in both men and women, right? These are tied to changes in the brain. So here's our follow-up 28andHe, where we can see these really beautiful AM to PM changes in hormones. And they have actually a little bit stronger influence on the functional connectome in the male. I don't show the data, but El Morada has a bioarchive paper that she put up last week showing the same morphological changes in cortical thickness, gray matter volume also in the male brain. So I think that's maybe sort of one thing that we can use to really debunk this myth that hormones only matter for the female brain, whatever the female brain is. No. These are powerful neuromodulators for all sexes and genders. Okay, what about other endocrine transitions, right? So this is a new precision imaging paper that's about to come out. We are tracking women starting preconception continuing about every 2 weeks across the complete gestational window, with follow-up scans going 2 years postpartum, right? Because if you think about the changes, the endocrine waves that happen during the menstrual cycle, well, that's dwarfed by the changes that we know are happening across gestation. What is wild to me is that human neuroscientists have exquisite maps of fetal brain development going up to about 24 weeks gestation. We do not yet have a map of the maternal brain. And all you'd have to do is move the bore of the scanner down 2 feet to get that. It's wild. So this is, there's been beautiful work from our Spanish colleagues and others looking at the brain typically pre and post partum. And here we offer sort of that sort of missing internal window. So how is the brain changing during gestation? And I will just say, we see sweeping changes in cortical thickness and gray matter volume. You can see here over time, so gray matter volume, which is showing this sort of reduction from pre-conception to gestation, and then a rebound in the postpartum period, again, going up to about 2 weeks or 2 years postpartum. There's concomitant changes in the ventricles and cerebrospinal fluid. We can look at this in a bunch of different ways, and I don't have time to do that now because that's not the point of this talk, but suffice it to say that, this is an incredible time for neuroplasticity in the human brain. And you can see this, just these changes unfold literally week by week. It's not limited to cortex. We see these changes happening in the cortical brain, subcortical regions as well. And the bioarchive paper is out, and the paper itself should be out too. So it's clear, right, that matrescence is this extended period of neuroplasticity in the adult brain. And at least for, the animal folks have known this forever. Humans are always catching up, human neuroscientists. But it's now, we can't ignore the evidence that pregnancy is just this incredibly powerful time. And what if we can harness that level of neuroplasticity and use those, once we understand the mechanisms that drive that, use that in clinical settings when we want to induce neuroplasticity. To do that, we got to take women's brain health seriously. I encourage you to read, so this is a postdoctoral fellow of mine, Magdalena Martinez Garcia. She just put out a really nice commentary on how we see the field moving forward, specifically within the neuroscience of human pregnancy. Okay, so that's small n, right? Big data. So how do we use precision imaging and collect lots of data, but in small sample sizes? But consortium studies are sort of the typical big N, big data, right? So big N, big data versus small N, big data. So let's follow the other path for a little bit. It's going to take a lot of time to make up for 30 years of lost science. And we find ourselves with a pretty unique opportunity to sort of buck the trend. So in November, we launched the Ann S. Bowers Women's Brain Health Initiative, and our mission is to close the gender data gap, right? To fill the void, to peer into that thin black hole of a line and make it our entire universe. Simply put, our goal is to take women's brain health prime time within the brain imaging community. So we're driven by a pretty radically simple idea that progress in neuroscience will flourish when the health of men and women are valued equally. And we're fueled by a commitment to make neuroscience inclusive, both in terms of who gets to ask the question and who is served by the answer. So this initiative was born out of another observation. Our current model of siloed science is failing us. Under the status quo, we have independent researchers who work in isolation from one another. We have brain imaging consortiums who churn out, data for these typically small scale studies, and then the data often stay disconnected. But this practice has led to underpowered data sets and pretty serious questions about replicability, and it is becoming clear to those of us in the human brain imaging community that this lone investigator model of science might be a dead end. It also overlooks the sort of single most important feature of at least the University of California, right? We are one university with 10 appendages sprawled out across the most demographically and geographically diverse state, right? We're the 5th largest economy on the planet. So what happens if we remove, right? We break down those silos and we sort of rewrite the rules of how science is done. What if we pool data being generated across these brain imaging centers? That's what we're doing. We have 7 that are being onboarded right now. So the basic premise of the Bowers WBHI is that we take brain imaging data that's coming in from each of these centers. We pool them together into a central repository. It's paired with a harmonized reproductive health history questionnaire, and then that data gets sent to our data repository. This is the first, what we call sort of the WBHI base model that can allow us to answer so many questions, right? Just by working together. Our back of the envelope calculations are that the University of California system alone generates data from about 10,000 unique participants every single year. So we can get big data pretty quickly if we just work together. I will say that I think, you always have to be inspired by, looking around at other fields, and we're not like the first to invent big data. You've heard examples within the neuroimaging community, but I'm always inspired by thinking about like astrophysics and genomics, right? They really demonstrated this power of embracing big data. And we know that like particle physics, right? If anybody's seen the documentary that just came out about the Large Hadron Collider, right? Like, look what can happen when team science is the thing to lead the way, right? Those team science approach ushered in major discoveries for those fields, and we can do the same for neuroscience. So, we're sort of fools not to. Okay, at the Bowers WBHI, we do 3 things. Sorry, get so excited. We generate the missing data, we build the infrastructure to share that data, and apologies for the text. And we are generating and building AI tools to really make sense of this wealth of data. And we have 3 funded centers. So the administrative core is housed at UCSB. We have our data coordinating center at Stanford, and we have our AI core that's housed at Cornell. And I want to give a shout out to some important players in making this happen. So Russ Poldrak is the director of our data coordinating center, and then Nina Milan and Amy Kusieski are co-directors of the AI Center of Excellence. We're also building out our biofluid analytics core, which will be housed at UCSF. So from the beginning, our goal has been to pair the best data with the best methods and the best minds. Suffice it to say that this is a deeply collaborative effort. This is a more fun way of thanking people. It requires engineers and data scientists and, flipping, and MRI technologists and neurologists and epidemiologists and women's health experts all working together, right? This is the opposite of siloed science. So a big thanks, this is from our launch in November. These are all of the people that actually get to, that make this work. I just get to stand up here and tell you about it. So thanks to them for pulling the bandwidth together. Okay, and I'm running out of time, but what about the data, right? As science, data is our currency. It's the thing that does the heavy lifting and solves all of our problems if we gather it correctly. So the Bowers WBHI really strikes at 3 levels: by going broad, but by going deep and going precise. So I will quickly walk you through what I mean by those. So we are detecting, as I mentioned, sort of in this our base model, pooling data across the University of California system to answer basic questions about women's reproductive health history and how that relates to brain health. Here's a commentary we wrote about one example and how big data can approach questions like oral contraceptive use in the brain. We also thinking about the importance of deep phenotyping, right? So the brain does not exist in a vacuum. We need to understand how sort of if we can collect data from proteomics, from biofluids, from wearables, how all of this can inform us about the health of the brain. And finally, this idea of precision imaging, right? We don't want to collect just a snapshot on the brain, but really understand how the brain is changing as it moves over time. So all 3 of these really sort of inform the sort of tenets of the WBHI. And in all cases, the ethos of the Bowers WBHI is that once the data are in hand, we simply give it all away. We collect it and then release it open access. to the global science community so that the discoveries never stop. And I won't go through all of this, but we have been so grateful to really sort of join forces, first and foremost with Cornell Bowers College of Information Sciences. They're conducting some of the world's finest research in AI, and I think joining our entity with theirs is going to create synergy that is much greater than the sum of their parts. So again, what do you do with this wealth of data? Well, you release it to the world, but you also sort of make sure that you have the computational scientists in place that can help you make sense of it. And this Center of Excellence is, one thing I want to call out is their organization of AI Grand Challenges. So every single year, our hope and goal is to release an AI Grand Challenge themed around women's brain health. Every year it'll be different, and that's led again by Amy and Nina. And really tapping into the AI community. So at the first Grand Challenge, they're hitting 10,000 different computational scientists that don't normally like work on women's brain health, but can use their talents to really help us make sense of it all. And I don't have time to get into any of this, but I do want to just say, I give you two examples of subsidiary studies that the WBHI is supporting now that we have this infrastructure in place. And one of those is led by Rachel Buckley. It's called the Longitudinal Menopause Project. And I won't have time to play this video, but we'll share it later. And our objective is to launch one of the largest studies in the world to date on menopause, and understanding prospectively why this midlife period is such a critical one, with deep phenotyping markers. And our second major subsidiary study is led by these three women, Liz Kraskell, Susanna Carmona, and Ina Stelzer, is the Maternal Brain Project. So to really, again, get a big data approach paired with precision imaging, so we can establish normative trajectories in maternal brain plasticity and also understand the sociocultural and biological drivers. And with that, I'll just say it's very easy to get angry when we think about the status of women's health, but there is so much to be hopeful about. As I mentioned, we witnessed the launch of the first ever White House initiative on women's health research. Earlier this year in the United States, a bill was introduced to bolster support for menopause research. It's not a done deal, but it's a start. We have editors at the top journals that are being supportive. And final plea is that I think we need to begin establishing universal standards for the brain mapping community by working together. So my group, and many of you will be tapped for this working group, have a draft of a lifespan women's health history survey that we can all begin to collect, so we know it's harmonizable data. A shout out to Elvisha Dhamala, who's doing the same thing for sort of a standard sociodemographic survey. So we hope that those can be released so that the brain imaging community everywhere, right, global, can make sure that we have harmonizable data. And with that, thanks to the team who's doing all of the hard work. All right.
One question. Where's the first hand? Looking, scanning the room. But come on. Okay, well then I'm gonna ask a question. No, I'm gonna ask one. I was just a little curious about the precision medicine. I have a comment first actually about just finishing things off with IRBs and REBs about issues around scanning pregnant women, because it's still, there's still like language and standard consent forms about that. So just putting my plug out there so people like yourself who are influential can educate our ethics boards. But the point would, or question would be around precision medicine and setting clinical cohorts. So beyond, I think, the methodological strategy, but my sense is in order to draw conclusions about people suffering from illness, you need to study people suffering from illness. But how do you feel about that sort of comment / question?
Yeah, I mean, I think precision imaging, precision medicine, you know, you have to think about the ethics and whether these people can give consent, but I think that is the step forward, and thinking about prodromal state. So part of our goal with the Maternal Brain Health Project is to enroll a large population of women so that understanding that about 10 to 15% of them will go on to experience perinatal depression. What are the first, like the earliest biological signatures of that we can map? Maybe it's in the proteome, maybe it's in the immunome, maybe it's in the brain, but like we need both precision imaging and big data to answer those sorts of questions. And I think that's the only way we're gonna begin to get answers to these questions. So thank you all
very much for a wonderful presentation. Okay. Right, and we now have Dr. Marianne Seney from the University of Pittsburgh who's gonna finish things off.
All right. Hi, everyone. Thanks again to the organizers for inviting me here. I've just had an amazing time. I think the vibe in this room is so exciting, and I'm so excited to the collaborations that we started today or this week and moving forward in the field. So I just have one disclosure. My lab receives funding from Arrivo BioVentures. So I wanted to give you just a bit of an overview of what we do in the Seney lab, because I think it will focus what I'm going to talk about today. So we use very much we're a translational lab, and in our lab, what that means is we do work in both the postmortem human brain in the context of psychiatric disorders and addiction, and we also do work in animal models, mostly in mice. But we're very much a multi-omics discovery-based lab. So we do these multi-omics discovery-based approaches in both mice and in human brain. I'm going to tell you today just about transcriptomic studies that we do, but we also do tons of other things I just don't have time to tell you about in various aspects of proteomics and epigenetics. But then what is something that's really important to what we do is cross-species data integration. So I have my scientist here. She's doing lots of data crunching. We do a lot of this in the lab. And these are very much hypothesis generating. And where we can then go into our mouse models and manipulate things at the molecular level and look for effects on behavior and integrate these into brain and behavior mechanisms. And ultimately, what we really want to do is be able to design more effective therapeutics. And for what I'm going to tell you about today, specifically drugs that might be sex specific. And we have two main arms of the lab. I'm going to tell you today about our work in major depression and in sex differences, but we have a whole nother half of the lab that's interested in something else that is different between people. And that's looking at circadian rhythms and sleep and effects on psychiatric disorders and addiction. But for another time to tell you about. So today I'm going to be talking about major depressive disorder. So these are facts that I'm sure we all know. Depression is highly prevalent. So just in the US, it's about 19 million people every year are affected. And the World Health Organization considers it to be one of the leading causes of disability and years of productivity lost worldwide. And unfortunately, the death by suicide is quite high in the depression population, about 15% lifetime mortality. And despite decades of research on depression, we have very few new pharmacological treatments in the last 50 years. But what I want to focus today on is sex differences in depression. And these come in a couple of different flavors. So first, we all think about this sex difference in incidence, right? So we have women are about twice as likely to have a single episode of depression and about 4 times as likely to have recurrent depression. But we also have sex differences in the clinical phenotypes associated with depression. So first, women are more likely, on average at least, to have depression where they have hypersomnia. And actually, on the flip side, men are still likely to have sleep disturbance. They just have insomnia. We also have females are more likely to have overeating and weight gain, and men are more likely to lose weight if they have depression. We also have sex differences in co-occurring disorders, with women having a really high prevalence of co-occurring anxiety disorders. In fact, these two often go hand in hand in women. Where on the flip side, men are more likely to have a co-occurring substance use disorder. And these are really interesting, these co-occurrences, and it potentially reflects different coping mechanisms in men and women. So we really use these clinical features here to ask some, I think, important questions. So do these sex differences reflect different biological pathways on the way to depression? And might they suggest sex-specific brain pathology? And if we understood what that pathology is, could that inform potential sex-specific treatment development? So if we think about this potential sex-specific pathology, we have a couple of different alternative hypotheses here. So first, it's possible that men and women will just have the same changes in their brains when we look at them at the molecular level, but maybe they're modulated by some sex-related factor, say gonadal hormones. If this were true, what we might see is that we would see similar pathological changes in the brains of men and women with depression, but maybe just women have larger effect sizes, for instance. But it's also possible that the pathology is different, right? And that we would see distinct molecular changes in the brains of depressed men and women. And, you know, we really think this is quite possible because of the fact that some of these actual symptoms experienced and the co-occurring disorders are different in depressed men and women. But in either case, knowing what this pathology is, we think is super important because it could potentially help us develop novel and maybe even those sex-specific treatments that are so necessary. So great, we were interested in looking to see if there are sex differences in the molecular phenotypes of depression. Well, where might we look? Well, we can take advantage of decades of research that point towards this cortical limbic network of mood regulation that's altered in depression. So just briefly, we have the basolateral amygdala. This is this limbic brain region that's critical to sensing and assessing emotionally salient stimuli. So this bottom-up control of mood. We have the dorsolateral prefrontal cortex, the DLPFC, this cognitive assessment of emotional salience. And between these two, we have the subgenual anterior cingulate cortex that really integrates this top-down and bottom-up control. So we're interested in looking in these brain regions at the molecular level. Well, how are you going to do that, right? Well, I'm really lucky to be in the University of Pittsburgh in the psychiatry department. We have one of the most well-regarded brain banks in the world for psychiatric disorders. It's been around for about 35 years. I'm on call on the brain bank a certain number of days a month. So I get to help dissect the brains when they come in. So this is what a fresh brain looks like, right? Like I was a neuroscientist for a long time before I was dealing with fresh human brains. I just think it's interesting. We look at the dorsolateral prefrontal cortex, subgenual anterior cingulate cortex, and the basolateral amygdala. So I'm just gonna, these first studies that we did, these came out a number of years ago, is said, okay, are the molecular changes that happen in depression in these brain regions the same or different in depressed men and women? And cut to the chase, they're different, right? So this is some stuff that Georgia had shown earlier as well. So there are plenty of genes that are differentially expressed after correction for multiple testing in depressed men and women. They're just different genes. And at the same time, or right around the same time, some work came out from Eric Nestler's lab reporting basically the same thing. And so that was like, okay, phew, it's real. And not only that, but these findings were consistent across cohorts. These aren't the same subjects. In fact, all of our brains are from Pittsburgh, and all of these brains are from Canada. So across countries even, this is true. So it really suggests that this is a real phenomenon. And so this was really exciting replication. But then we said, okay, well, maybe we really need to understand what these small number of genes are that are affected in both men and women and with depression, because maybe if we understand what those are, we can develop treatments for both men and women. However, 75% of those were actually changed in opposite directions. So we're like, okay, well, we have to rethink our analysis here. So what we did is we performed a more rigorous analysis here, and we performed a sex by disease interaction. And even after, again, correcting for multiple testing, there were over 1,000 genes that were had a significant interaction of sex and disease. So we said, okay, interesting. Well, let's probe the data in a different way. And for this, we used another statistical approach called rank hypergeometric overlap, or RRHO. And this is threshold free, right? So it's not just saying like, what are the genes that reach some arbitrary level of significance, and are they the same in men and women? But what we do instead is, this is just what the interpretation is here, and we walk you through it. So what we do is we take all of the genes that we analyzed in depressed men and women, and we rank them. So this is about 12,000 genes. We rank them, so on the x-axis in men, to the genes that are most significantly upregulated, those that are relatively unchanged, ranked down to the ones that are most significantly downregulated. We do the same thing on the y-axis with women. And what you want to do when I show you these plots in a second is look for hot spots in any of these four quadrants, right? Because this would suggest that genes that are changed are similar in depressed men and women, or opposite, or we could see just like no enrichment at all. And what we see is using this threshold-free approach, this is highly statistically significant. We see that the genes that are affected in a threshold-free manner in these cortical brain regions are really opposite in depressed men and women. Then we said, well, what are these genes that are affected in opposite ways in depressed women? Well, they seem to load onto two different categories here. They're related to synapses and neurons. So these genes are significantly decreased in depressed men and mostly increased in depressed women. And they're related to immune function. So we have pathways and microglia. I know we've heard a lot about microglia these past couple days. These genes are significantly increased in depressed men and decreased in depressed women. So this really suggests that there's sex differences in synapse and immune-related changes in depression. So this was all kind of novel. So we want to do is say like, okay, wait a second, how does this jive with the literature? Does this even make sense? And we think it does. So first of all, up until the time where we were performing these studies, people were just doing postmortem studies on males, or they would throw a few females in there, but you could never really look for sex differences. And what we see at the transcriptional level is completely consistent with those male studies, at least in male subjects. And it's actually also consistent with rodent studies. So if you take a male rat and you expose it to stress in cortical brain regions, you get reduced dendritic complexity and increased activated microglia. So it's really consistent with a model in which in men, at least, what seems to be happening is that we go from these reactive microglia could potentially participate in excessive pathological removal of synapses. But what we see in women, at least at the transcriptional level, it suggests the opposite. And although that was surprising, it's consistent with the rodent literature. So if you expose a female rat to that same exact stress exposure, it actually in frontal cortex will increase dendritic spines and decrease activated microglia. So consistent with this sort of model. And really, I want to emphasize this. We really think what's going on is that we have sex differences in just healthy control individuals. And when disease comes around, they converge on a similar phenotype. And this is what happens in rodents. It's also kind of consistent with what we see in our data. So if we just took all these genes expressed exclusively in microglia and we look at them across the 4 groups, we actually see that the sex differences in controls. And when MDD happens, they converge on intermediate phenotype. So all of this that I've told you so far is all expression of genes at a bulk homogenate level. So we just take a chunk of gray matter and we chop it up and we look at gene expression. But more recently, we've more sophisticated approaches. So we're looking at expression in individual cell types. And I just wanted to show you just a little flavor of that because it's hot off the presses as of like 2 weeks ago. So this is where instead of just chopping up all of that and seeing what gene expression is, what you do instead is you look at expression of individual cells, and then based on those expression patterns, cluster cells. So in an unbiased way, then you can say what kind of cells that you're looking at. And then a first pass at this analysis, we just took our control subjects, although we're doing this in MDD and or just haven't done the analysis yet, but just in control males and females, we asked, are there any genes that are differentially expressed within any of these cell types? And the answer is yes. And by and large, they're mostly microglia specific genes. And these are all corrected for multiple testing as well. So again, just right back into, really suggesting that they're microglia specific sex differences in just control subjects, and then really understanding what happens in disease. I think we have to understand what's happening in just normal males and females first. So all this is great. We're doing work in postmortem brain. We think it's really important. It's the disease organ of dysfunction and psychiatric disorders. But all of this is correlative, right? Like we don't know if anything that we see is causing depression in our subjects. But we can take advantage of the fact that the cortical limbic network is very highly conserved across species. And because of that, we can then perform studies in our mice. And so basically what we're asking in our mice is, can we use mouse models to understand the sex differences we observe in humans? So where might these sex differences come from? So in just normally developing individuals, we see that it all comes down to their genetic sex. So if you have a Y chromosome, you will develop testes based on the expression of this SRY gene. They'll release testosterone, and during various sensitive periods of development, typically around third trimester and in puberty in humans, we see that these can cause organizational effects of hormones, which can cause more permanent changes in the brain and behavior. We also see activational effects of hormones, but I really want to focus today on these sex chromosome specific effects because I think that they're really interesting and very much understudied. So just Georgia told us about the FCG mice earlier, so I'm going to just go quickly through this. So in our FCG mice, this is our strain that which we can use to say, can we isolate effects that are due to genetic sex that are decoupled from gonadal sex? And otherwise, we can't do that in normal wild type mice. So we're going to call the XX mice are pure females, the XY minus SRY are pure males, and then we have these interesting combinations in between. And so then the next question is, okay, well, we have our model, what kind of stress exposure are we going to use? And I think that it's important to consider what type of stress exposure you use and which type of sex difference you want to model. So if we wanted to model a sex difference in a clinical phenotype of depression, then we want to use a mouse model that elicits a depressive-like behavior in both sexes. And we've done that using unpredictable chronic mild stress. If you're interested, you can look at our paper. But today I wanted to tell you about this work that we've been doing with Georgia Hodes, looking at, trying to model sex differences in the incidence of depression. And she told you about this model, this 6 days subchronic variable stress. And her lab has shown that females are vulnerable, but males are resilient to this stress exposure. And we think it's really interesting. We recapitulate in our just pure males and females, the female susceptibility, and we can see that it's exclusively driven by the genetic sex of the individual, and seems as though gonadal sex does not affect the stress susceptibility. But then we're really interested in what might be driving this, right? So we know what happens, but is it what is happening at the transcriptional level? So what we did first is we looked to see, these are kind of very similar to what I showed you in the human studies, right? What are the genes differentially expressed in females? What are the genes differentially expressed in males? And we see, yeah, right, they're different, right? And even when we use this threshold-free approach, so I'm reminding you that you want to look for hotspots in any of these quadrants, actually it's basically no hotspots at all. They're just like totally different effects of stress. And the pathways are also quite different as well. And in males, we see predominantly effects on immune-related genes. But I want to remind you that these males are actually stress resilient, right? They did not change their behavior. So actually, we think that it's possible that some of this programming and these vast genetic changes that we see might be promoting resilience. And finally, now we have these 4 core genotype mice. Well, what's driving this sex-specific effect of stress? So we can use this RRHO again. What we do here in just the females, we first on the x-axis, we look for the effect of stress in the pure females, and look on the y-axis to see if it's recapitulated by XX genotype or by having ovaries. And you can see that this is much stronger signal here, suggesting that the female susceptibility might be predominantly driven by their genetic factors. But the opposite is true in males. So the same thing here, the effect of stress in males on the x-axis, the XY genotype effect on the y-axis here, and the effect of testes here. And this is very strongly driven in the male resilience is driven by their having testes or hormonal factors. So just to summarize, the second part here, I hope that I've convinced you that there's more to sex differences than just circulating hormones, and that we have to consider genetic sex factors as well that are independent of hormones. We see that the stress susceptibility is driven by genetic sex, and that at the gene expression level, both genetic and gonadal sex are influencing gene expression. We're doing a lot more in this realm, getting, again, just like we did in humans, much more specific. So we're looking at transcriptional profile within microglia, specifically in the context of stress. And then we're using various immune and microglia-specific manipulations to see if this affects stress susceptibility in a sex-specific manner. And with that, I just want to say thanks to current and former members of my lab, collaborators at Pitt. So I want to give a big shout out to Georgia Hodes, who's I showed you some of our studies today, some of the members of my lab and funding. And again, just a special thanks to the family members who generously donated tissue of their loved ones at a very difficult time, without which this research would not be possible. Thanks.
Go ahead.
Thank you for this great talk. So I wanted to ask you, what do you mean by genetic difference, right? Because it could be XSKPs. It could be, I don't know, the X inactivation and what it actually has by itself, a lot of repressors and stuff. It could be some Y-linked genes.
Oh,
Because
it's so,
the
yeah.
genetic difference at the end,
It all
that
comes
sounds-
back to that, though.
Gene expression. And another thing that I wanted to say, I really love this 4-core genotype model, but I told you we have the data that show that there's interaction between gonadal hormones and X chromosomes. So, and this is also another aspect of it, like, so I was just like thinking, what is when you look at your gene expression data, do you see some changes in SKPs or X-linked gene expression, or how would you explain this if it is not through gonadal sex?
Right, yeah. So, I mean, I definitely, like, what we can do is I specifically isolate the effect of genetic sex. That's like a main effect, right? Or the main effect of gonadal sex. But you're totally right. There's definitely interactions of the two. And that's true. I mean, we've known that for decades, right? Of, neuroendocrine research that, if there's interactions of these factors, we're trying to distill it down to like, can we isolate any of these? But when we analyze our data, we're always looking at those interactions as well. I just find them a little trickier when, because under normal circumstances, an XX individual is not having the hormone exposure of a male, right? And vice versa. So it's a little bit more tricky to think of like the clinical relevance of it, but it's true though, yeah.
Might have pretty much similar genetics, right? So it's not like a genetic sex in terms of, oh, there's a variability in genetic variability. It's something about what this X chromosome does or a Y in cell. Is it gene expression or is just the presence in the
Oh,
cell?
yeah, we don't
What
know.
about that?
We don't know. And maybe we can talk more because this is like right up your alley, right? Because like we haven't gone beyond this because there's so many different things. Is it like having multiple, the 2 X chromosomes? Is it having a Y? Is there some interaction with autosomes? Like, we just haven't gone that far yet.
Can I ask a question that
I'm not
might?
sure.
Yeah, no, I got permission.
Yeah.
It's very quick. So I think it's brilliant, lovely. I of course think that hormones are going to be related in some cases. In your 4-core genotype model, did you only do the 6 days? Because
No.
I wonder if, because obviously MDD wouldn't be.
Yeah. So I just didn't have time to talk about it. We published this a few years ago, where we used the unpredictable chronic mild stress in the 4-core genotype mice and basically did exactly the same sort of studies. I was just going to talk about this because it's a little bit newer, but yes, in fact, so when we did that, we saw that the behavior was driven by both hormones and genetic, and it depended on which behavioral test you looked at, which one it was, and the same thing with gene expression. So like some genes that were affected by, the sex difference was driven by hormones, sometimes it was driven by genetic. So yeah, we did that.